Welcome back, everybody, to our afternoon session. I hope everyone had a delicious lunch. Our first talk for the afternoon session is by Jennifer Possley at Fairchild Tropic Botanic Garden. And the title of her talk is Going with the Flow, 30 Years of Fairchild Tropic Botanic Gardens, Rare Plant and Habitat Conservation Actions and Research, with a focus on the Deering Estate Flowway.
Thank you. Can you hear me okay?
Yes, perfect.
All right. Hi, I'm Jennifer Possley. I'm at Fairchild Garden. I'm the Conservation Program Manager. And I think, surprise, Vanessa, I decided to share my time slot with Lydia Cuni. So I'm going to, the first half of my talk, I'm going to kind of give an overview of the work we've done over the years at the Deering Estate. And then Lydia is going to focus in on our recent work in the Deering Estate Flowway. So we will get started. Let me see if I can share the screen. Vanessa, can you see the PowerPoint okay? Okay, thank you. There we go. All right. So we are going to be talking about Fairchild's work over the last few decades at the Deering Estate. And we work with a group of, I think we're 5 staffers plus numerous volunteers. And our program goal is to reduce the extinction risk of rare flora of South Florida. So we tend to go places where there are rare plants. And we, of course, spend a lot of time in the Deering Estate. It's the most botanically diverse preserve in all of South Florida, as far as I'm concerned, outside of Everglades National Park. There are more than 500 native plant species, and 117 of them are considered to be rare by one metric or another. So we're out there very frequently, sometimes almost weekly, and in partnership with staff from NAM and EEL and Deering, having fun, getting dirty, collecting seeds, collecting data, and monitoring. And this is something that we have been doing since about 2002. And I'm going to talk about our rare plant monitoring program that began in 2002, our rare plant reintroductions, and a couple of our long-term projects. So what happened in 2002 was Fairchild and Miami-Dade County signed an agreement to have Fairchild staff start a rare plant monitoring program for all of the county preserves. And that has been renewed over the years and is still going strong today. The work we do at the Deering Estate, we now have long-term data sets for population counts, and in some cases, more detailed data for federally listed species like the 4 you see here, the prairie clover, the deltoid spurge, the Carter's flax, and the tiny polygula. And we'll mention that through our monitoring, the 2 on the right, the Carter's flax and the tiny polygula, were actually rediscovered at the Deering Estate after having thought to have been extirpated for a couple of decades. Fairchild found the Carter's flax in 2014, and Dallas Hazelton found the tiny polygula, I think, in 2002. Although we haven't seen that one again in several years, so we're waiting for hopefully a prescribed burn. This guy will return from the seed bank. We do work in the hammock as well as the pine rockland, and we have monitoring data from Carter's orchid, the brown hair comb fern, and the grid scale maiden fern, to name a few. And then we also, as part of our rare plant work, we work on reintroducing and augmenting some of the extirpated and very rare species at the Deering Estate. So this is a list of the projects that I'm aware of. And you'll see that the last 4 all happened within the last year or so. And that's entirely due to the efforts of Vanessa and Dallas, who really have just been there for everything being on site to check on plants and watering plants so we don't have to. And that partnership has really been what's made these introductions so successful. And I'll just give you a very quick overview of each one. Fairchild and Deering planted 190 crenulate lead plants in Deering's North Pineland in 1995. So this is, at this point, this is a very old reintroduction study. It's one of the oldest in the United States. And it's been successful. There, of the 190 plants, there are 31 surviving, and at our last count, 199 recruits or seedlings. That number of seedlings goes up and down through time, and it definitely responds well to fire. So this is another one we're hoping to get to see fire in the pine rocklands at the Deering Estate. The grid scale maiden fern, we put out 211 in 2014. And this has been a very successful project as well. The survivorship has been 68 plants, which is pretty good for a fern reintroduction, and they've already begun recruiting on their own. Now we get into very recent history, and the brown hair comb fern, we put out 26 plants in 2019, and then we had some really dry weather for the next following months, and Vanessa and Dallas just really did a lot of work watering these plants, and all 26 of them are still alive. They don't all look great. Some of them look amazing. Some of them are still kind of on the fence, but this has been, so far, all signs point to success with this one too. The marsh is Dutchman's pipe. This is kind of Dallas's passion project. He obtained the seeds for this plant, which is extirpated from the Deering Estate, and he got them from the next closest population, Biscayne National Park. And Rose at the Deering Estate especially has been taking care of these plants. And as far as I know, all 7 are alive. And the Deering Estate is Vanessa and Rose are collecting seeds, giving them back to Fairchild so we can use them in research and seed banking and to grow more plants. So that's been cool. If you come on the Deering Estate field trip tomorrow, you might see one of the 4 modest spleneworts that we installed last year. They have to have protective cages over them because critters like to pull them out of the rock where we install them. And then our most recent effort has been run by Dr. Sabine Wintergerst, who is Fairchild's Seed Lab Manager. And this has been an effort to introduce the coral hoary pea to the Deering Estate. And this was a seed introduction, which usually when people do reintroductions, we put in whole plants, but here we decided to try seeds. It's gone really well so far. And the reason why we're doing this is this plant is only known from one site in South Florida, and that's the USDA at Chapman Field. So we're trying to reintroduce it to a nearby protected site like the Deering Estate where we know it's not going to be developed and it's going to be taken care of. So with this one too, all signs point to a successful reintroduction, although it's still very early. And I just wanted to mention too that we have over the years conducted several long-term monitoring projects. We have pine rockland plots that are permanent and still out in the estate. They were installed in 1994. And we also have, for a separate study, some monitoring transects that were installed in 2008. And these all have iron rebar marking the transects so they can be resampled. There's been publications from both of them. The plots were published in 2008 and 2018, and the transects in 2014. And I put up kind of my favorite results from each study. From the pine rockland plots, this is kind of a not very interesting graph. It's showing that there's a weak relationship between the size of the natural area and the richness of native plants. But that's what I think is so cool about it, is that some of these teeny little preserves in this study, of which Deering was one part, can have really high native species richness. And then the study that looked at the pine rockland transects, where we compared the management status of pine rocklands all over the county. This is a study where we came up with some recommendations for vegetation cover that's now used as a standard in Miami-Dade County land management planning. So things like litter depth should not exceed 3 centimeters, and you should have at least 25% cover of native grasses and herbs. That's where that comes from. And then, of course, we have long-term monitoring in the hammock and wetland. We actually started this in 2002 at the request of the Parks Department, Natural Areas Management, and we installed a study looking at the vegetation adjacent to Cutler Creek, and we wrapped that up in 2016 and switched to a plat-based method in cooperation with the South Florida Water Management District. And then I'm going to turn it over to Lydia. We're going to see if we can work this out so that she's narrating the rest of the presentation, and I will be manning the slides for her. So take it away, Lydia.
Thanks, Jen. Hey everyone, Lydia here. So I'm going to be talking about the vegetation monitoring that's happening in the hammock in the wetland areas going on past 2016. So in the map on the left, you see some black lines along the Cutler Creek, which are the transects that Jennifer was referring to that were monitored since 2002. And in 2016, 6 10 by 10 meter plots were installed following the South Florida Water Management District ecological monitoring plan. And these plots are pretty much proxies for what's happening in the larger communities. So there are plots in the hammock, in the transitional sort of hammock, in the slough, and in the mangroves. And the plot design is nested, so you can capture the different layers of vegetation. And from this data, we can have the species richness and diversity of all the plants. We take the herb, shrub, and canopy coverage of all plants. We measure the tree seedling abundance. And from all this data, afterwards, there's a floristic quality calculation, which is quite interesting. It's based on giving a plant a conservation value and averaging this for the plot and coming up with a floristic quality index for each plot. And some other data that's collected is water depth in these plots, and also photo point monitoring, which you'll see later. So some of the data quickly, through the years from 2016, 2020, here's a graph of the richness in each plot through the years. So the transitional hammock in plot 153 has had the most richness, and this does include some non-native species, just be aware. But there could not be very many, maybe at most 5 to 7 non-natives in a plot, and over mostly native species. And the least rich is the mangrove plot, which bounces between 3 and 4 species. And in general, you see that the plots experience the highest richness in 2018 following Hurricane Irma. And it's just an interesting pattern to observe. So looking at the coverage that these trees take up, the basal area coverage of tree species, on the left, we have 2016 coverage by species, and on the right, 2020. And things are pretty stable. One thing to note is that there was a large recruitment of, here, deep purple, this salix, the Carolina willow, large recruitment and so it increased the basal area, the tree coverage in the plot. And something to note is royal palms are staying very stable. And in plot 153, the hammock plot, just some loss of a big tree make a difference. That light purple is a gumbo limbo, which we'll see in a photo later. Here, looking at, instead of by species, by the total tree basal area coverage in the plot. So the mangroves had the highest basal area. There was quite a number of trees that were tagged greater than 5 centimeters in DBH, and something in 2020, many of these trees no longer had leaves, so we counted them as dead, and so that's why you see the basal area really drop off for that species. But the other plots are relatively stable, and you see in red the increase of that that willow coming in and increasing the basal area, the tree coverage. Yeah, so out of 100 meters squared, the most that the trees cover right now is, over 5 meters squared coverage in the plot. So looking by plot through the years, you can see again that each plot is pretty stable in the number of stems that were tagged, where the biggest increase was those willows in plot 399. They were now larger than 5 centimeters, so they were included into the plot. And on the right, the mangrove plot, which had a sharp decrease in the number of stems that were tagged. That's an interesting die-off of some mangrove trees. And looking at the seedlings, the woody seedlings that we measure in the plots through the years, the hammock plot has the most seedlings, the woody seedlings throughout the years. And overall, there was an increase in 2018 following Hurricane Irma. And that could be from lots of light entering the canopy and having seeds germinate. And so we recorded a great number of woody seedlings that year compared to the other years. So looking at the photo point monitoring, this is the hammock plot 153. And on the left, you can see that large gumbo limbo. And the understory is kind of sparse. You can see right through all the stems. And on the right is this last year, 2020. And that gumbo limbo died and fell down. And that's what was that loss of that basal area. And you can see the understory, the shrubs are starting to grow up, and there's more green coverage in the photo. Next plot on 2016 on the left, there's a lot of vine coverage, not much shrubs. And the vegetation here in this plot is quite dynamic because this is one that the water levels go up and down quite a bit. And so on the right, you see that it's more open. This on the right in 2020, more open, and just very little cover in terms of shrubs. Just big royal palms and royal palm fronds. And another picture from the mangrove plot on the left in 2016. You can see the understory is pretty much just pneumatophores and all the stands of a black mangrove on the left there that you can see in the picture on the right. The black mangrove is still there, and you see in the understory lots of mangrove red mangrove seedlings coming in, see much more green. And something to note is just a little bit of a green sheen in the water. There was some filamentous algae that we observed this year. You see that coming in. So with that, that's my overview of the last years in the flowway, and thank you to everybody for listening. And if you have any questions, please ask us.
Thank you so much, Lydia and Jennifer. We do not have time for questions. If we can address any questions that the audience has during the panel session. We're going to move on to our next talk, which is by Alan Frank, Fungi of South Florida, Diverse and Virtually Unknown. I have made you a co-host, so you should be able, oh, no, sorry, just kidding. It is Mason Bradbury. Sorry, Mason. Let me find you really quick.
So while you're transitioning, Vanessa, I just want to do an additional shout out to Jennifer and Fairchild. I missed the first part, so I don't know that if you acknowledged that Jennifer and the Fairchild Tropical Gardens science team were one of our champion partners of the year award this year for their long-term collaboration in ecological monitoring. So I just want to share that honor as well with the group and others. So, and Mason's one of our fellows. Yay.